Hello, I am Fernanda, and today I'm here to present to you how to interpret image-based profiles using single-cell retrieval and single-cell visualization through Google Colab. So in this presentation, we are covering this paper that we published recently that is called Interpreting Image-Based Profiles using Similarity Clustering and Single-Cell Visualization. And in this paper, we have instructions as well as the GitHub repository associated to it, where we provide instructions for the researcher or the user to look at their image-based data and try to interpret them and have insights about how their treatments, how their compounds are clustering, and what this means biologically at the end. And for that, we propose two basic protocols. So the first one is using Morpheus, this software that is a web-based tool where you can explore similarity matrices, as well as a notebook on Google Colab where we import the data, process the data, as well as plot the single-cell visualization and the field of view visualization as well. So for the first basic protocol, based on Morpheus, we already have two videos available on the COBA YouTube channel. And on those videos, we explain the basics and the concepts related to morphological profiling on the part 1, and on part 2, we explain and show how to practically explore this data on Morpheus. So if you want to have more details, you can read the publication as well as watching both videos. So on this presentation and also in this demonstration, I'm going to show you how to use our notebooks with real data. And it's important to note that on the paper, we have an example data set that is available on GitHub, but you can follow that through the instructions of the paper. But here, I'm going to show you a real world example using my own data. So interpreting image-based profiles can be a challenge, and it is a challenge. That's why we have tutorials and the publication. And processing the data before the interpretation is an important step. So normalizing the data, annotating the data with metadata, as well as doing feature selection. These steps are very important. So we also have those on our notebook. And so this notebook will help, of course, with processing the data as well as retrieving the single cells that are representative or random. So for that, we are using Google Colab. Google Colab is a hosted Jupyter Notebook service, and it needs no setup to use, and it gives free access to computer resources. So that's why we are using Google Colab. It is important to show you how the data set looks like. So this is a data set that was pre-processed and analyzed using cell profilers. So we ran some pipelines to extract the information from the images using cell profiler. And this is the data set that we have. And here are some metadata information that are important for this processing. So for example, the name of each well in the plate, the name of the sites, and the file names. So we need to know the name of each image as well as the width of the image, because we need this information to make the single cell crops. And we also have, of course, all the features related to cytoplasm, cells and the nuclei as well. Okay, so, and as you can see, the names of my channels, you also need to know the names that you gave to your channels. So for example, here I have Core GFP and Core PI that are the names of my channels, and these are also important to know. So we can plot the single cells for each channel that the images were acquired. And as you can see, it's also like a single cell data frame. So you have each row would be one cell. So this is the GitHub repository where all the informations from the paper are available. And here we have as well the link to the Google Colab notebook. So you can click here and open in a new tab. And as you can see, you need first to do a copy of this notebook to your own Google Drive so you can edit it. So you're just gonna make a copy. And then you can see there is a summary of what we do in this notebook. So we import some specific libraries, define some inputs. We can import a data set that needs to be processed or a data set that is already processed somewhere else. We also need to choose the information to plot, calculate some correlations and some visualizations, and at the end, we can visualize the single cells. So the first step is to run these 3 cells to import the necessary libraries. So as you can see, we are cloning our GitHub repository where we have some Python functions that are all necessary to run this notebook. And we also have some pip installs for specific libraries that we are using. And then finally, we import those libraries. So this step will take about 5 minutes. So after 5 minutes, now we need to define the inputs to run this notebook, and you can read more about each input here. And if you are running using the example from GitHub, you can run this cell and it will take care of the inputs for you. But in this case, we are running an external file. So the first thing you're going to do is to run the cell, and it's going to ask to mount the Google Drive, and you need to grant permission. I already granted, so mine already says that it's mounted at Google Drive. And if you come here, you're going to see your Google Drive. And now you need to find where your data is located. So you can come here and find your data. So the first input are the profiles, which is the table that I just showed you with the features and the single cells. And the first thing we need to give is the name of this file. So you can just copy and paste here with the CSV. Then we need to give the path to this file. Okay. Then we need to provide the path to the images. Since we are using only images from one plate, we need to give the path to the plate. But if you have a data set where you have images from different plates, images that are located in different folders, you're going to give the main folder, so the images folder. You wouldn't need to give the path to the specific folder, because later we are going to deal with that if you have images in more than one folder. But here I only have this plate, so I'm giving the path like this. And then it's going to ask you if you need to annotate your dataset with an external file. Say yes or no. So we are actually using an external file to annotate our data. So we're going to answer yes. And we need to provide the path to the metadata. So here I have metadata from this experiment. And I need to provide the path. So I just come here and copy the path. And I also need to provide the file name. Just come here and copy that. And then we need to give the names of the channels that we have in this data set and that we want to plot. So if you're using a cell painting data set, for example, you're going to have DNA, RNA, ER for the names. But for this data set, I have core GFP and core PI. And also we need to know the prefix for each column that specifies the file name. So if you're using data that is not from Cell Profiler, this name can be different. So you just need to provide this. But ours is from Cell Profiler, so it's like file name underscore. And here I need to give just one column with the width of the image so we can crop the cells later. So you need one column that has the information for the width of your image. And if you're using a compressed image, you should say here, but in our case, our images are on TIFF file, are in TIFF. And then you need to provide the size of the crop. So we are saying that 150 are a good start, but if you see that it's cropping too much, or yeah, it will depend on the size of your cells, actually. So you can change this later. And here we are importing the data set to normalize and feature select using this notebook. So in this case, I'm running that. And we are going to import the data set. And we, of course, need to answer about the number of plates. So as I was saying, if you have more than one plate in this data set, here you're going to have each folder with the name of the plate. And you're going to have to say in your data frame, where do you point to the name of this folder? So usually the way that we have, it's a metadata plate column with the names of each plate. And the name of each plate will be the same name of each folder. So in this way, we know where to look. So if you have a representative image or a representative cell being located inside in one of the plates, you know where to look for the single cell. But ideally, you would have only one plate here, so it's more manageable. Okay, but we have this option if you need to. I'm gonna say no. And here we are importing the metadata file that we pointed and annotating our data set. So as you can see, this is a good troubleshooting to show you. We are using PyCytominer to perform these operations, and we need to specify what are the columns in our metadata file and also in our dataset that will be used to perform this merge. So I can show you how the metadata looks like. If we come here and we download this, you can see that we have the well position here. And as I was showing you on our data set, we also have a metadata well column in our data set. So you're gonna have to change here depending on how your metadata file are organized and how your data set as well. So if you have different names, you're gonna have to change here. Unfortunately, it's manual. You need to just open here. So if you click twice, or if you go just here and show code, you can come here and change. So for the first one, we have the well position. So inside our metadata file, we have the well position column. And PySatominer actually is going to add the metadata in front of each column. So that's why I'm putting metadata well position here. And this one, it will be the one on my data set. So mine is just metadata well. So if you run this again, now it should work. And it will annotate our data set. So yeah, it works. So you need to pay attention to that. And then here we are just going to do the normalization. And to make the normalization, you want to select the columns that are representative of the metadata. So the columns of the file name, the metadata, and also the width of the image are going to be kept before doing the normalization step. That's why we run this cell. And here we are going to choose which sample is our negative control. Because in normalization, you can usually do normalization to the negative control or to all the samples. So this is just a choice that you have here, and you can read more about it in our in a post that we have in our blog that I'm going to also link in the description to understand which one is the best for you. But here we need to choose the negative control, and I can choose the metadata compound and then choose which compound is representing my negative control. In my case, it's the non-treated cells. So the cells with not treated with any compound. And then you save that. And I'm asking, is there more than one plate in this data set? If yes, choose the column where the plate information is. But in our case, we said no, that we don't have. So it's gonna say to proceed with normalization. And now we need to normalize, so we can normalize to the data set, to the negative control, as I was saying, or per plate to all the samples. So in this case, I'm going to normalize the data set to the negative control. And this can take a while or not. It depends on the size of your data set. And then we are doing feature selection on this data set based on, again, on PySatominer. And we are using correlation threshold, variance threshold. We are dropping NA columns. And yeah, we are using these operations from PySatominer that you can check on their documentation, how they are performed. But these are the usual pipeline that the lab uses. So I'm going to run this, and it's going to take a few minutes to run. So feature selection here, it took about 7 minutes to run this cell. And this is normal because it's a big data frame, and it's going to tell us how many columns it removed, and then we can just join back some columns that we had filtered before, and now we can create a new column. So if you want to modify this, or if you don't want to run this cell, that's okay. It's just a way to have the names of our compounds and the concentration in just one column. And that makes it easy later. And of course, here is just an option if you have a data set that was already processed, that is already normalized, already feature selected, you can use this one. And so here, in these next steps, we are going to choose the metadata information and the compounds that we want to plot. So I'm choosing the new column that I created, the compound concentration. And you can choose to plot all the samples that you have or just a few selected ones. I'm going to plot just a few of them so we can see, and it's better to visualize here, but you can just run this 4.2 and you will plot all of your all of your treatments. So let's choose one more. That's okay. So we are plotting these 4 compounds and we are saving them. And then you can also use this notebook to generate a similarity matrix plot. Of course, you are going to do that if you follow the protocol from our paper. You are going to do that in Murphy's, but you can also run the similarity matrix here, and you can export this figure if you want. And you can see that some compounds that I have, they are more correlated than others. And yeah, it's just another way to visualize your data. You can also save this correlation plot and download this image. And in this section, in 5.1, we are going to create just a column called correlation that we are going to add to our data frame. So we have the values that we have here in our data frame. And it's just a way to compare our compounds to some baseline. So for example, if I want to understand how my compounds are correlated to the non-treated, I can use that, and I'm going to create a correlation column that's going to have, so here I can show you correlation 2. So this is the value of the, for example, aspirin 1 has a 0.7 correlation to the non-treated cells. And the same for all of the compounds. So this is just a way to have this information in the data frame if you want. Of course, you can choose not to use this information later, but it's useful if you want to compare your treatments to a baseline. And you can also visualize this kind of the same information, but in a table. So for example, I select the non-treated ones, and I'm going to display the top 5 compounds that are closest to the baseline treatment. So for example, based on that, I can have a sense of the compounds that are more similar to the non-treated, and therefore they can it can indicate that these compounds doesn't have have a profile that are more similar to the non-treated. It's just a different way to look at this data. And then finally, we can visualize the cells. So we are going to do a random or a representative selection of those cells. And here you can choose the method. So you can choose random or representative. And then you can choose the correlations. If you want to use this correlation column that we created to order your image plot, is just to see the cells ordered in a way where the first is the baseline that you choose, and the next ones will be the closest treatments to your baseline. And of course, you can also choose yes or no. You can try different ways. And then we can choose the number of cells to plot for each group. If you choose more, it's gonna plot the single cells, the representative cells, 2 or 3, for example, one below the other on the plots. You can also try that later. And if you run this, now we are going to select the cells from the original data frame based on being a random or representative selection. As you can see, it didn't take too long. It was like 42 seconds. And now we need to do some image path manipulations on the data frame so we know where the images are. You can choose if you want the image intensities rescaled or not. We also have more information about that on the published manuscript. And here you need to provide the pixel size of your images, and it will depend on the microscope and on the lens that you used. So you need to check this information with the manufacturer. But so in our case, this value is 0.35 micrometers per pixel. And then now we are going to plot the images. So the first plot that we have is just the images in a shuffled order and without the labels. So it's like trying to have an unbiased way to look at your samples. So trying to observe and see how for example, here we have the DNA and the RNA in this channel, and here we have the acidic vesicles on the PI channel. And as you can see, for example, for this treatment that I don't know which one it is, you have these vesicles distributed and more concentrated in this area of the cytoplasm. Meanwhile, in this one, you have less acidic vesicles, they are more sparse. You can also have different distributions of the nucleoli in this case, more intensity in this cell, less intensity in this one. So you can start thinking about the biology and what does this mean for your data set. And then we can add the labels and see how which cell is which. And yeah, as you can see, I have the non-treated, some cells treated with aspirin, others treated with orphanodrine, different compounds that leads to different phenotypes. You can also plot based on the correlation. As I was saying, I chose the non-treated cells as the baseline, and then you have the cells that are closest to the non-treated based on the correlation plot. You can save the image and download to your computer. And you can also plot the images based on the name, so A, D, and O. So based on order by the name of your compounds. And finally, you can plot the images that corresponds to each representative cell. So then we can see on the field of view how those cells are distributed. So for example, in the aspirin, you can see that we have less cells and they are more intense in with this dye that we used. And yeah, you can have a sense of how the cells are distributed across your field of view. And then finally, you can save also this image to your computer and use later for a paper, for a publication, and you can see our manuscript as well as yeah, giving feedback to us and saying if you need any help or if you want to make some suggestions. You can also use our GitHub for that. You can submit an issue saying if you need any help with this notebook, for example. Thank you very much.